Well, it's more complex in the sense we're only looking at the genetic risk, right? And there'll be other risk factors for many diseases have many other clinical risk factors. Like, for example, in cardiovascular diseases, obesity or type 2 diabetes are well-known clinical risk factors. And just by having those conditions put you at high future risk for cardiovascular disease, right? There's also a strong genetics component in cardiovascular disease too, but you need to be able to either add them together or say, I already know this patient's at a certain risk, but he or she's at an even greater risk because they have a genetic component, right, to the risk. To your point, Dr. Stanford, it's very important to be able to keep in mind all of the other risk factors that pertain for any particular disease.
Welcome to the Precision Medicine Made Simple podcast, where we simplify the complex world of precision medicine. We're here to explore how personalized healthcare can empower you to understand and improve your health to live a longer and healthier life. Join us as we break down the latest in medical science into actionable insights. Let's make precision medicine accessible and actionable for everyone. This episode is sponsored by Biography Health, where DNA-powered healthcare is designed just for you. Biography Health is a HIPAA-compliant precision medicine platform that equips providers with AI-driven recommendations powered by your genetics, health history, and clinical insights, all while keeping data private, secure, and independent from large EHR systems. If you're a provider ready to bring precision medicine into your practice, visit biographyhealth.com to learn more. Or if you're a patient who wants your doctor to explore this, point them our way. We'll help them bring personalized healthcare to life. As a reminder, this episode is for educational purposes only. This episode is in no way a replacement for professional health advice. Please consult with your professional primary care provider for medical advice. If at any point you feel that a topic discussed on our podcast was misrepresented, or if you have additional insights that could enhance our discussions, we wholeheartedly encourage you to reach out to us. Your feedback is invaluable in helping us improve and in ensuring our content remains both informative and reflective of the latest developments in precision medicine. Hey everyone, welcome to the Precision Medicine Made Simple podcast. I'm joined here with my co-host, Dr. Stanford. And for today's guest, we are talking about a very particular topic. Well, number of topics here. But in today's world of genomics and precision medicine, the biggest challenge isn't scientific possibility. It's representation. Too many datasets and predictive models are built on narrow ancestries, leaving billions of people underserved. So our guest today has spent his career changing that. With an MD and PhD from the University of Chicago and training at Harvard, he went on to co-found Decode Genetics, published nearly 200 papers, and helped scale platforms that connect genetic discovery to real-world medicine. Today at Galateo Bio, he's leading the charge to build a global, diverse biobank that could transform precision health for everyone. It is my pleasure to welcome Dr. Jeff Gulcher. Thank you so much for coming onto the show. And just to kick off this episode, from my understanding, I'm just so curious how you and Dr. Stanford connected. How did you guys get connected in the first place?
Well, I think it was when we were trying to develop better tests for different ancestries with polygenic risk scores at Galateo. And we were looking for physicians that had a strong background and experience with human genetics already for their patients. And certainly Dr. Stanford fills that criteria perfectly. And he was interested in what we were doing as well. So we decided to band together and see how our tests might work and how they might improve with Dr. Stanford's feedback and expertise.
What drew you into working on PRS scores in the first place then? Because I understand you have a personal story that's happened with you and your father, but then was there interest even before that? Like, how did you even start to just get fascinated into that field?
Well, it really brings us back to our Decode days, or even at Harvard Medical School. I did my PhD and did some research with Kari Stephenson, who at that time was a neurologist, neuropathologist at University of Chicago, then Harvard. And then we co-founded a company together called Decode Genetics, based in Iceland, because one reason Kari is from Iceland, but Iceland also has some great features, at least in the early days of human genetics, because the population is relatively homogeneous. It's not homogeneous, but it's relatively homogeneous, so it makes it easier to find genes or at least variants above the signal, a stronger signal above the noise that you might get when you're working first with multiple ancestries at once, right? Like in the United States or other places in Europe. So it was a great starting point. So early on, our ambition was to eventually genotype and sequence most of the Icelandic population. There are about 350,000 Icelanders there. And so we were able to sort of create sort of an industrial scale genetics laboratory, right? And that was really facilitated by the Icelandic population because they were eager to be involved in the studies, mainly because Dr. Stephenson, one of the most popular people in Iceland, really drove this vision. So we set this lab up in Iceland, and then eventually we would genotype large numbers of patients who consented to be in our studies with 500,000 or so genetic markers, and that was sort of the rise of what are called genome-wide association studies. And that was really started back in about 2005 for us, 2004, 2005, where we were able to get DNA chips that Illumina made. It was probably closer to 2007 when we started using those full scale in Iceland. And what it does is it just leaves no stone unturned across the genome. You have 1 billion letters that you have to deal with. If you're really going to measure everything in the genome, but there are only certain variants, certain letters that change across the population. So you can first simplify the task by just defining or genotyping, sort of DNA fingerprinting with maybe about 500,000 variants instead of 3 billion, measuring 3 billion bases, right? And so we were able to do that. And then what we did was we would compare maybe a few thousand patients with prostate cancer versus everybody else in the population that doesn't have prostate cancer, and look to see, are there variants, these genetic mutations that are more common in patients with the disease as compared to those who don't have the disease. And so that's just an association study across the genome. Back in those days, most of the genetic work was really looking at families, right? Looking at large scale breast cancer families, for example. And that's where BRCA1 and BRCA2 were initially found by looking at big chunks of data, chunks of the genome across the family, and looking to see, are there certain chunks of the genome that are inherited that correspond to the patients in the family that have the disease, right? So that was just genetic mapping, linkage analysis in the early days. After that, though, with patients who don't really have a family history, you're sort of stuck. What are you going to do to discover genes driving the much more common diseases that really don't have a strong family risk? And so that's where association studies came to be, where you just simply measure whatever your favorite gene is in a bunch of patients and compare that to a bunch of individuals who don't have the disease controls, right? But around the time that we did the first genome-wide association studies, people were just looking at their favorite candidate genes. So it used to be called candidate gene association studies, right? And so you just pick your favorite gene based on biology, right? And you ask the question, okay, let's look at the variation, and does it correlate with the disease? So you had countless, thousands, tens of thousands of articles that would show these correlations, but usually were done with one gene only or one variant, and maybe only a handful of patients, right? 100 patients and 100 controls, right? And statistically, you're going to, if you do a lot of that, you're going to find some interesting associations. But the problem was, if somebody at Harvard published something, a different group in Stanford may not be able to replicate, or usually did not replicate those findings, right? Because they were really false positives. There wasn't statistical power really to be confident that association was real. And furthermore, you really want to look for the strongest associations across the genome, not just put all your eggs in one basket for a couple genes, right? And so you really need to be able to look at variation across the genome. So Illumina's DNA chips, once they got to about 300 or 400,000 variants, that finally put it within the range of being able to do efficient what's called genome-wide association. These are hypothesis-independent studies. We're not making a guess ahead of time which are the key genes that we should be looking at, right? And so that's where the discoveries came from, is now you let the genes that have the strongest effects rise to the surface, right, above the noise. And so that's what we started with. There we're looking, like I said, a few thousand patients versus maybe 100,000 individuals who don't have the disease. and then found some of the first genes, for example, for prostate cancer. So once we found 8 different variants that we've been, by the way, it was very important to replicate these findings, because is this just an Icelandic thing, or does it also replicate at least in other European populations? So we were able to get a large number of prostate cancer patients who had consented for genetics research from the US and Europe. And one of the sites that we worked with was Bill Catalona's site at Northwestern in the United States in Chicago. And he's famous for being one of the most, accomplished and prolific prostate cancer surgeons. So he had a very large patient base that were willing to consent for research. So he would send us his blood samples or DNA samples for replication. So he was on all of our papers that we published, with these initial findings. These are the first genes for prostate cancer discovered. But what's interesting, though, is these genes were a little bit different in character, or quite different in character compared to the BRCA1 or BRCA2 genes that you find in family-based studies, like for breast cancer, BRCA1 and BRCA2. Because there, if you have a very rare mutation, you may have a 5-fold increased risk for breast cancer in your life, right? So that's the example of a monogenic high-risk gene for cancer. In contrast, for the 8 genes that we found, usually if you had the variant, you'd increase your risk for maybe 1.2 to 1.3 fold, right? Increased risk by not 300%, like in breast cancer, BRCA1 and BRCA2. It would be more on the order of maybe increased by 20% or 30%. But we found that when we combined all 8 of these gene variants together, then there were some individuals in the population that would be predicted to have at least a 2-fold or even a 3-fold risk for prostate cancer, just on the basis of aggregating or combining these genes that each had minor effects for prostate cancer. But when you combine them together, there's a certain percentage of the population, maybe 5 or 10%, that have at least a 2-fold risk for prostate cancer. So that's why we decided let's combine those variants together and see if they were more useful or could be useful clinically.
Right. So is that a simple summation of those risks, or is it more complex than that?
Well, it's more complex in the sense we're only looking at the genetic risk, right? And
Right.
there'll be other risk factors for many diseases have many other clinical risk factors, like, for example, in cardiovascular diseases, obesity or type 2 diabetes are well-known clinical risk factors. And just by having those conditions put you at high future risk for cardiovascular disease, right? But there's also a strong genetics component in cardiovascular disease too, but you need to be able to either add them together or say, I already know this patient's at a certain risk, but he or she's at an even greater risk because they have a genetic component, right, to the risk. So, but to your point, Dr. Stanford, it's very important to be able to keep in mind all of the other risk factors that pertain for any particular disease. In fact, those risk factors can be modifiable, right? You can modify your risk due to obesity by trying to lose weight, right? If you're successful doing that, right? That's a perfect example, right? Where there are other risk factors that are not modifiable. So your genetic risk really isn't, in most cases, modifiable per se, unless there's a drug that can tag the pathway encoded by one of those genes, right? But it is still useful to define what is your risk. So even if, for example, a lot of individuals who have cardiovascular disease, heart attacks or strokes, they don't have clinical risk factors. They don't have diabetes. They don't have obesity. What were those risks? What was their risk, right? In many cases, it's this genetic risk that many times does not get measured.
So it sounds like it's from the genetic risk standpoint for polygenic risk scores. You're pretty much let's just pretend that there's 10 risk alleles and 10 that are protective, and you have 8 of the risk alleles, but only 2 of the protective, and so that somehow gets summed together.
Right.
From my understanding, and I'm looking for clarification here, there's not more complex logic applied. For example, if you have 2 particular risk alleles that occur together, it's more than additive. It's
right. Yeah. So the probably the best model still to this day is a multiplicative model, which, some call it a additive model because it's log additive, but a multiplicative model where you're just simply multiplying these risks together. So if I had your example, let's say I had one variant that doubled my risk for prostate cancer, but I had another variant that decreased my risk for prostate cancer by a factor of 2. Those in principle would neutralize each other, right? And when you multiply 1/2 times 2, then you're sort of back to neutral, right? All other factors being equal. Obviously, it depends on the frequency and other things too. So, so what we do nowadays is we don't just restrict ourselves usually to 8 variants like we did when we first discovered and created a test for prostate cancer risk back in 2008 or 2007. Instead, we look at maybe thousands or tens of thousands, and some models have even more than that in the genome. And so you're trying to aggregate them. In most cases, you're still assuming that they're working independently. So that's why it's still more of a multiplicative model, if you will, or sort of multiple.
Right. It seems like if there was a consideration of the actual function or loss of function that occurs, if it's present here as a risk allele, but there is a protective allele over here, or not a protective allele, it would be far more complex of a problem, but more predictive if you could find predictive patterns, as opposed to just, randomly adding or
Right. And it's, it's not so much random, but I know your point is that, well, you're randomly measuring these and you're trying to include them all in the model. There's different gradations of it. One is, well, you just take the most significant variants, which is what we did in the early days as well, right? And you still see that in some models, though, like the latest breast cancer model really only has that Myriad uses also in their polygenic risk test only has, I think, 88 variants for breast cancer. These are for the common variants, right? Not BRCA1 and BRCA2. And they get very good performance, where others believe that you should try to bring as many variants together, but you're still assuming that they're working independently and they're aggregating and you can bring them together. The problem has been, so some have suggested, well, maybe this variant and this variant interacts, right? And as you suggested, right, the risk is even higher than simply multiplying the 2 risks together. And the search for those, when you have so many different variants, right, is very, very tough because really you're just sort of you're increasing the number of tests.
I mean, the computation capability and the challenge of the machine learning to find that.
Right.
But it seems like that would be the holy grail, so to speak.
Yeah. And so a lot of groups are trying to work on that, looking for patterns using AI for that. But so far, it looks like the models that are so far working the best in real world clinical data, still seem to be the multiplicative models.
Yeah, it's just amazing. It works as well as it does.
I have a question when it comes to PRS scores, because when I noticed, there could be a discrepancy if you get it from one company to another, right? Like from my understanding, one, I guess, how is there a discrepancy then? I mean, it sounds like it just could be modeled differently and trying to identify a different pattern. But if somebody was to then try and vet which PRS score to listen to, how would they go about doing that? Is it then looking at where that data was pulled then and looking at the ancestry and what that population data is? Or
there can be multiple or many differences or many reasons for why you get different results, right? So one can certainly be that they didn't use enough. Maybe they used a much smaller number of variants compared to others. It seems that the trend has been that as you add more and more variants, these models seem to be more stable, at least within the European ancestry population, because most of these discoveries have come from European based populations, right? European ancestry populations, whether they're from the United States, studies in the United States, or studies in Europe. So if you make the discoveries in a certain ancestry, they don't always translate. So that's another reason why the PRS model based on one ancestry may not work as well for those who have other ancestries. And it doesn't mean that you have to have, you can just have a subtle difference in ancestry, right? Where you can have maybe 20 to 30% East Asian ancestry and still have 70% European ancestry. The model can be off compared to those who have mostly a European ancestry. So that's been a big challenge, I think. And that's why I think PRS scores over the years have got a bad rap because you can easily point out, well, these are not consistent across populations, right? But they seem to be pretty consistent within the European ancestries, right? The conventional models, right? And to the point where, for example, obviously I'm predominantly European ancestry. We made the initial discoveries in Iceland, and all the models that we had were based on European ancestry. And my relative risk myself for prostate cancer back in 2008 was about almost twofold, about 1.9 fold. And I was just looking just now to before this at my latest profile on the latest and greatest model that 23andMe now uses as of about a year ago for prostate cancer, and they incorporate an awful lot of variants, right? Many more variants than 8, right? And my relative risk is almost identical today with 23andMe measuring my DNA from my blood or from my saliva, quite in a different lab than what I originally had my profile done in Iceland, right? And of course, our models were quite different, and we had fewer variants, right? However, the variants that we found first, right, the stronger variants are the ones that are always easiest to find with smaller numbers of patients, right? So those 8 genes have some of the strongest, even though it's modest still, 1.2, 1.3. Those variants are taking a good share of the risk, defining a good share of the risk, right? Whereas a lot of other variants that were found along the way when you combined different data sets together. So now instead of having 3,000 prostate cancer patients, you might have 100,000 prostate cancer patients. Then the variants are very, very modest, 1.02 or whatever, that kind of thing. So I think there is consistency, but the ancestry has been the problem, right? It's not. I think it's less so the models now, since most models are built upon a very large number of variants that have been replicated before, or they've used these linear logistic regression models that combine everything together and let it drop that way. The ancestry has been the one where they don't translate as well. So there have been several companies, including our own. You have Genomics PLC. You have several others, Lilica, and other groups, and myriad of themselves, right, trying to make adjustments of their polygenic risk scores for certain diseases based on ancestry.
So you mentioned ancestry being like one of those like factors then, but what about testing methods? Because you mentioned 23andMe. I mean, they do genotyping, but if I did, let's say, whole genome sequencing, could then I get more accurate PRS scores from?
Well, that's a great question. You just imagine if you're doing DNA chip typing, with let's say 1 million variants, right?
Right.
Then the average person has about 5 million variants that are different from a quote reference genome, right? On average, 4 to 5 million, right? So you're measuring about 1 million, but that's usually stacked towards more common variants, right? The DNA chips have been built that way, unless you make it customized. And so from that perspective, you're measuring very similar variants across different platforms. And even if they're not measured exactly the same, you can impute the variants in between. And typically your models are based more on the common variants that are common across many patients. They're not the individual, very rare variants that you might see in a BRCA1 or BRCA2 gene that has very high risk. Now, if you say what's different between whole genome sequencing and a DNA chip, well, so a DNA chip that's doing a good job for common variants that are at least present in 1% of the general population, for example, right, or maybe even higher than that. Versus for whole genome, as you suggest, hey, you're measuring every single variant, right? You have difficulties with some structural variants, but in general, you're trying to measure everything, whether it's rare or common in the population, it's present in the patient, right? Or the individual you're sequencing. Well, if your models depend more on the common variants, right, you're not really gaining anything extra from the very low frequency variants that your models probably aren't going to include, or if you're trying to include them, they're not going to have a big impact compared to the more common variants. So from that perspective, if you do a good job sequencing, or you do a good job of DNA chip typing, which most groups can do very, very well, then there shouldn't be a big difference across the platforms. Now, 23andMe, they do a great job because they've used Illumina chips since day one. And those are very, very accurate, right? There's some other chips that may not be as accurate, but nowadays, most of the chips have very, very high reliability on the variants that are actually, encoded on the chip itself. So,
did you ever consider another population outside of Iceland? Like, were there a couple other populations that came into consideration just based on?
Yeah, when in Iceland, when I was at Decode, we used Iceland. We worked with Iceland as the discovery population for the reasons I mentioned,
Right.
but very important to be able to replicate outside Iceland, as I mentioned. So we had about 500,000 patients from around the world, right? Asia, East Asia, United States, other European populations that had very specific and very well characterized disease populations. They weren't big populations like Iceland, but they might have 3,000 heart attack patients, let's say from UPenn or other places, and they would send us the DNA on their own patients, but we wouldn't do the full genome-wide measurement of variants because the chips were very expensive back in those days. Instead, we would just focus on the few variants that we had discovered, right? And if it would use their population as a replication set. So, but you're right. I mean, that's very important to be able to show replication across populations. But in terms of using other populations besides Iceland as a discovery population, since I left Decode back in about 2013, Kari and the team have continued to expand, much beyond Iceland, right? Certain U.S. populations, for example, in Utah, and some Scandinavian populations that they've been involved with. They've done an awful lot of genotyping and some sequencing in Danish populations and so on. So they've continued to expand the envelope there. And of course, they were also the ones that sequenced most of the UK Biobank. They sequenced the AGD, that's the Vanderbilt cohort of 250,000 patients that were put together by Illumina and Vanderbilt and supported by 8 different pharma, including Amgen and Decode. So, I think there has been this drive to get beyond certainly relatively homogeneous populations like Iceland, but I think it still hasn't swung to the point where we are where we need to be, I think. And that was the reason that Carlos Bustamante founded Galateo Bio in Miami, Florida, is to try to build up large-scale cohorts that had much more non-European ancestry. And that's what we've been doing for the last few years in Miami. So what we've done, and that's important because if you can do some of these same discovery efforts in other populations, for example, Latin American populations, right? In South Florida, there's a higher prevalence of, for example, Hispanic or Latino populations. And then we have a lot of collaborations in Latin America itself and South America and Mexico that where we're able to get consented samples and collaborate with those medical centers to try to make discoveries on those populations. We have cohorts also, collaborations in Taiwan to give us more of an exposure to East Asian populations. And then we have other U.S. sites that happen to be in areas with higher prevalence of African American or other Hispanic or East Asian populations. So that's very important because that gives you sort of the raw material to make discoveries of perhaps new genes that are not as strong in European-based populations, but are much stronger, or at least easier to find in some of these other non-European ancestries. And there's many examples of that where genes have been easier to find, let's say in East Asia, in Korea. And when we didn't see that gene in Iceland, but then we were able to replicate that gene because we were just focused on replication. So the bar wasn't as high to quote that it would be if you're trying to discover it for the first time. So there's many examples like that. And we hope to expand the number of discoveries that are made instead of continue to work with UK Biobank and some of the very good European dominated populations and just discover the same gene again and again, right? Try to go to other populations and hope to find even more interesting or perhaps different drug targets that might be easier to find there or might even be more specific for those populations. And that's going to be important as we try to expand medicine and drug development beyond U.S. and Europe, right, into other populations or those subpopulations within Europe and United States. We need to, make better drugs that work either more universally across different ancestries, or maybe you need to have certain drugs that work better in certain subpopulations. And the only way you discover them is one way to discover them is to use human genetics to help define what are some of the key pathways that you might want to consider for drug development.
I got a quick question for you, Jeff. When you're challenged about the legitimacy of PRS, polygenic risk scores, or what makes this ready for prime time, et cetera. Is there a single journal article that you would, the one or two journal articles that you would point someone towards that really details how polygenic risk scores are done and how it's been studied and legitimized?
Yeah, I mean, there have been several review articles. The only thing is there's been some differences in terms of what is required, right, to come up with ancestry, what we call ancestry-aware PRSs. So some believe that if you just simply bucketize a given patient into whatever their predominant ancestry is, that's enough. So, for example, if you're the average African American in the U.S. is about 20% European ancestry, 80% African ancestry. So if you took somebody who, let's say, was African American and maybe only, let's say, 60% African ancestry and 40% European, you would, using this approach, you would say, okay, let's measure the ancestry. Because this person had greater than 50% African ancestry, I'm going to treat them as if they're 100% African ancestry and put them in that bucket and so on, right? And the problem with that is, well, a large number of us are admixed, right? We have more than one continental ancestry, right? Most Hispanics have a lot of European ancestry, but they may also have a lot of African ancestry. You may have 3 or 4 major types of ancestry. And we're not even talking about subcontinental ancestries just with that, right? And so I think those approaches don't work as well as if you consider the full ancestry more carefully for a given patient to try to understand and correct the PRS. So and there's some other groups that do it as well. What we try to do is we try to look at your full ancestry. We try to make sure we have good resolution on that so it's accurate. But then what we try to do is we try to match your ancestry to the profiles that are probably most appropriate, the PRS models that are most appropriate for your mixture. We call it admixture, but it's just mixtures of ancestry, and try to find a better fit that way. And that seems to work out pretty well. It's not perfect, so I think we still have ways to go to continue to improve. That's why we're trying to build up these big data sets with other ancestries, but at least it allows you to be much more discriminative than before if you make those adjustments. And we have our own papers that sort of demonstrate that. But I think there's a lot of room for improvement, I think, on the algorithms. But I think also as we continue to build up these big multi-ancestry data sets, we and others, they'll get better and better as time goes on.
How would someone vet that these are bad polygenic risk scores that you shouldn't pay attention to? Yeah,
well, I mean, the tip-off usually is, we've demonstrated this in one of our papers, where if you start with a PRS or a set of models that are based on European ancestry, and then what you do is you look to see how do they perform in some of these other populations, right? So you need to have a data set, right, of, let's say, Hispanic or East Asian or whatever. And what you do is you look to see what's the distribution look like in terms of, risk. So typically you can imagine having sort of like a big Gaussian curve, right? You just have a big middle, right? So most people for PRS scores are dead in the middle, right? They have really no significant increased risk, no significant decreased risk overall, right? But then where the interesting things that happen, right, are patients that are actually in the upper high tail, right? They're in the upper 90 or 95 percentile in terms of risk, right? And there you can, those patients might have maybe a 2-fold or even 3-fold risk for coronary artery disease, right? Or high blood pressure compared to the general population. So what you can do is you can map that out going from one ancestry to the next. And if there's a distortion, either there's a shift in the curve, or if it doesn't look nice and symmetric, then you realize that you have a problem. And then the other problem is you'll get a flattening where you can't really distinguish between those in the 90 percentile versus the 10 percentile. There's not much difference in terms of the risk, right? So that indicates that a model that works really well in European ancestries, now it's started to flatten. It's not working very well in these other non-European ancestries. Then you have a problem. So we demonstrated that with a few different populations on our cardiovascular traits. when we publish that, you can actually see that there's this flattening when you use the model, the European-based model. But if you use our model where we do the ancestry adjustment, suddenly now you see the curves look much more discriminative, right? They do a much better job even in these other non-European ancestries. So that can give you an idea of whether or not you're on the right track. And then finally, you can look to see what's your prediction success rate, right? You're just simply looking at what's the predicted risk of a given set of individuals in a cohort, and is that reflected when you observe those individuals in that cohort? So you sort of put them into different buckets, and for example, if the patients that are in this bucket in the upper 10 percentile, overall you predict them to be, let's say, twofold risk. Well, you should have twice as many heart attack patients in that group compared to the general population. So that's the type of thing you can do to calibrate or assess and validate the PRSs.
So ancestry adjustment is really like the very specific change. Is there any population out there that you would say not like the most underserved, or there's just really not that much data?
Yeah, Hispanics in general, I would say the most underserved, because you have very large cohorts that have been supported by governments in East Asia, right? Especially Hong Kong and Japan, some in Korea, an awful lot going on now in Taiwan as well. And so from that perspective, you have some of these, at least DNA chip type, there's not a whole lot of whole genome sequencing that's gone on yet, but a lot of DNA chip type, which is really the only data that you really need for these ancestry adjusted models. So I think that they're well served. European, obviously, and of course, given a lot of the studies have been done, larger studies have been done in the United States, a lot of them do have a component of African American, right? But I think the biggest underserved is, Hispanic populations, right? Just because, they may not be included in a lot of these studies, even in the United States. There's not as many, obviously, in other populations like Europe, for example, and then in Latin America in general, there hasn't been a huge effort, right, in terms of doing large scale, nationwide studies like you see in the UK or Iceland or other places. And so, but that we're trying to rectify that at Galateo, right? We're trying to bring in resources, capture consented patients, work together with the collaborators and scientists and physicians there in Mexico and other places in Latin America to try to rectify that, but also leveraging the Hispanic populations that are nearby us as well in Florida, right? Because they have excellent medical records and they can complement what we also work with other populations.
I got another question. So, you mentioned admixture, and certainly we're rapidly getting to where there's more admixture than not. So how, I guess mechanically, mathematically, formulaically, do you handle someone that is half European, half, well, there you go. Okay. So half Filipino. How does the algorithm adjust for all these different combinations?
Yeah, you're going to have to ask our data science group. I don't know the precise details, but just imagine, though, if you had, let's say it was in your case, half, let's say East Asian and half European ancestry, right? And so let's say the relative risk of this might be from the European side might be twofold, right? But for the Asian side, right, if you used the Asian model, maybe the relative risk is much lower, right? And might be that same profile might only be, let's say, 1.2, right? Well, you could say, well, maybe it would just be the simple arithmetic mean between 2 and 1.2, right? Because in principle, that's sort of what is it? What's driving what the ancestry is driving a certain risk. If you have admixture, then you have two different ancestors that are sort of driving perhaps the same risk, but perhaps different risks, right? And so that's what you're trying to model with these. And we do, use AI algorithms to try to model the effects of the ancestry on the corresponding risk profiles.
So you're not necessarily just looking at a cohort of half European, half East Asian, and comparing to that. It's not that.
Well, it's certainly, that's very helpful though to do when you were trying to do validation work, right? To sort of confirm that your models are doing a good job, right? And so we try to do that as well, right? Trying to take, and you can also model too, right? You can model the different admixed populations, simulate, I should say, and then test your algorithms out on that. A lot of that has been done as well by others too. So I think it's all trying to get to the same level. Can we be more and more confident in these? Are they truly accurate and non-European ancestors? I think most would conclude that the European ancestry based models applied to predominant European ancestry appear to be, for whatever they're worth, right? They appear to be pretty accurate in terms of defining risk. Now, these are not diagnostic tests, so doesn't mean that if you're twofold risk for prostate cancer, you're going to develop prostate cancer. But does that magnitude of the risk, twofold or threefold, does it really match reality or not? And it seems that most of the models now for European populations seem to be pretty consistent, like I just mentioned with my own prostate cancer risk. But it's where I think the wild card still is on the non-European ancestry, and how deep do you need to go in terms of not just admixture like we're doing now, but also what about the subcontinental ancestry, right? When East Asia, we're talking about East Asia, that covers all of China, Japan, Korea, Taiwan, and so on, right? And is it enough just to put them in one block, or is it better to have that? So it'll be interesting to see, do the adjustments become even better with higher resolution of global ancestry assessment? And we're doing some testing on that as well.
Yeah, you need a lot of computational capacity. I think as that gets better and better, and AI, everything gets faster.
Yeah, I think you're right.
There's going to be an exponential, refinement, improvement, and accuracy. But certainly, I think we can all agree PRSs are in fact ready for prime time. From the standpoint of, can it add to the predictive value of what's going on? And absolutely, it does.
Because if you're just looking at, say, breast cancer risk, of course, everybody knows the story of BRCA1 and BRCA2, and there are many other POLB2 and some other high-risk genes. In the typical panel, I think Myriad's still using about 11 genes. There are some other larger panels too, but the strongest genes are BRCA1, BRCA2, and POLB2. Mm-hmm. That puts you up to about 60 to 80% lifetime risk for breast cancer, as opposed to if you didn't carry one of those variants, that your risk is, if you're European ancestry, it's about 12% lifetime risk. So very significant. And of course, that's actionable, right? Where it can be prophylactic bilateral mastectomies or earlier, more aggressive detection, et cetera, right? For those that are positive. So, but that represents probably less than a few percentage points of all breast cancer patients, right? Now, this is the number is increased percentage. Yield is increased in family if you have
women or something like that.
What's that?
I think it's like 1 in 400 women, or 1 in 400 people, I should say, have the BRCA gene.
Right, they have this rare variant, right? And then if you look at the breast cancer patients who have breast cancer itself, right, it's on the order of a few percentage points. It goes up though if you have a strong family history of breast cancer, 1 or 2 or 3 individuals, it keeps going up, and that can be as high as 10% in some series, right? But once again, you're focusing just on, if you're just to focus, and today you can only typically get reimbursement if you have a family history of breast cancer. But if you don't have a family history of breast cancer and you have not had breast cancer yourself, then it's hard to get a reimbursement for that test, right? Unless you want to self-pay or other. So, but there are a lot of patients that are beyond those who have family history, right, that are BRCA1 or BRCA2 positive, and we're missing those, right? But it's still relatively rare, right? That's where the PRSs come in. To your point, Dr. Stanford, was, there's another strong genetics component beyond these very rare variants of high risk for breast cancer, and those are PRS. Those can be represented by a PRS test, polygenic risk test for breast cancer, and so you get another 10% of patients that have at least twofold, threefold risk for breast cancer. In general, they're not going to have the 6-fold risk, right, of breast cancer, 5 or 6-fold risk of breast cancer that you might get if you had a single high-risk variant, BRCA1 or BRCA2. But still, would you consider at least earlier screening for those who are positive? Screen it or with mammography, or more intensive screening with breast MRI, right, which is not used for everybody, but for those at higher risk based on family history, or if they're African American, that it's more suggested to have that earlier and more intensive screening. So, is that the actionable part of it, right? Same thing for prostate cancer, right? So my own story with prostate cancer, when back in 2008, when we discovered those 8 variants that I mentioned, we had created our own PRS test, and we actually, even before we had used a DNA chip for our clinical testing, we just measured those 8 variants, right, with just a PCR based genotyping test, right, in our CAP-accredited laboratory in Iceland, and we would provide that to the United States and Europe in a variety of different clinics. But I took my own test shortly after we created that test, a validated test, and it put me at about 1.9-fold risk for prostate cancer. Now, my lifetime risk based on being European ancestry is about 16% to have clinically significant prostate cancer lifetime risk, right? So this put me at about 32 or over 30% lifetime risk, right? Now, that's not enough to say, well, are we, what are we going to treat or do biopsies right away and look for prostate cancer?
So So you found out about this elevated PRS risk before you were diagnosed?
That's right. Yeah. So I had the PRS test. Then I was only 48 at the time. The recommendations at that time, and still really to this day, if you don't have any family history, risk factor for prostate cancer, screening should not occur earlier than age 50, right? So the recommendations usually are start screening with PSA and digital rectal exam periodically once you become 50 years old, right? Because prostate cancer is an older person's disease, right? But you can certainly have an onset earlier than 50 years old. But if you have family history, in the guidelines to this day, it's recommended that you get earlier screening with digital rectal exam just to see if you can feel an abnormal prostate, and also PSA, right? It's just a blood test.
So that's what you went for at that time at age 48? Yeah.
So my PSA was at 2.5, which today is considered abnormal for somebody at age in their 40s, but it was considered borderline abnormal then. And on repeat, it was still 2.5. Went to Bill Catalona, who, as I mentioned, he's the urologist head of urology at Northwestern, and he looked at me and said, "Well, he didn't feel anything on exam, so which is not unusual, right? Because most prostate cancer you can't detect by rectal examination." But he said, "I don't think you have prostate cancer because you're too young." I mean, 48, that's pretty young, right? For prostate cancer, most prostate cancer occurs after age 65. So, but he said, "Well, let's just go ahead and, given your genetic risk, given that you have some indication something might be going on with this elevation in PSA or borderline elevation in PSA, let's go ahead and biopsy." So he did 12 core biopsies there. And
did he do an MRI first or just went straight?
Back then, there was no such thing as MRI imaging for the prostate back in 2008. But, or if for those that were doing,
there was no organ-specific MRI with contrast.
No, there was the images back then were totally useless because they were so fuzzy, right? They're much better now, right? But that required an awful lot of tinkering, right, to get to with the protocols. But so anyway, so all you could do really is just to do biopsies. So I did 12 core biopsies. 3 out of the 12 cores were positive for a Gleason 6. That's sort of an intermediate grade prostate cancer. 3 out of 12. But given that's 3 out of 12, that's more territory involved, and more importantly, this was clearly multifocal because it was on both sides. The prostate has 2 lobes, so it was on both lobes. The lesions were just on one. So from that perspective, and also given my younger age, that puts you at higher risk for eventual spread, right? And so I did have a prostatectomy, radical prostatectomy with nerve sparing that he performed. And then after that, I checked my PSA. My PSA went down to 0 as expected, right? And then after that, I should mention when they looked at the pathology of the prostate, it's about 15% of the prostate had cancer, and it was Gleason 7. So, which is common because a lot of times the biopsies aren't very representative of what the pathology is. So it was a Gleason 7, which put it at a higher grade than on the biopsies. And since then, I've had 0 PSA, so it hasn't referred.
That was a miraculous sequence for you. Probably saved your life.
So I think so. And then my father I forgot to mention, I did have a family history. I do have a family history of prostate cancer. My father had it, but not until about age, I think, 68. Sort of later onset or usual onset, late in life. Prostate cancer is not really a strong risk factor for early onset prostate cancer by itself. And but he had a complete resection at age 68. And what's amazing is almost 30 years later, he had a recurrence of his prostate cancer, not from the original tumor, presumably, but from whatever was left after he had done the surgery. Some prostate tissue is left, not with tumor, but it was considered a secondary primary. But so potentially aggressive form.
So I think there's some good testing coming out for minimal residual disease, and blood test monitoring, biomarkers, circulating tumor DNA. I mean, that's going to be coming like light speed. And along those lines, let's say, and I have this situation all the time, I have patients whose Galateo report says they're 2.1, 2.2 times the population average risk, ethnicity, and gender adjusted. Obviously, for prostate cancer, you don't need to gender adjust it. But what's the recommended? Does Galateo make recommendations in the reporting? And if so, what's the recommendations for someone at high risk, other than monitoring PSA and PSA velocity and density and all that?
Right. And earlier screening, or what we say, we try to be careful here. We say patient may benefit from earlier screening, right? But even most of the guidelines suggest that you talk directly with your physician to decide when is it appropriate to even screen with PSA, for example, right? Because different guidelines have different sort of age ranges, right? What's the maximum age or what's the minimum age, right, for screening, and making it clear clearer. What would you do if somebody had a positive PSA, for example? And it's not the best, it's not a perfect test. So how do you manage that and those expectations with your patient? So we try to be careful about that. But it is interesting. There was a paper in New England Journal from NHS in April, and there PSA is not very well used in the UK. Because of the lack of good sensitivity and specificity from their perspective, so it's not as well used. But what they did was they looked at a cohort. They consented everybody ahead of time, and they said, okay, let's go ahead and run a prostate cancer PRS, right? Mm-hmm. And so they decided, they had already pre-specified that they would offer doing biopsies and MRI. Well, they did biopsies and every MRI on anybody who had an elevation risk based on, I think that was the top 90 percentile. So if you're at high risk based on PRS, they would also offer surgery, regardless of what the MRI showed, right? And
surgery or biopsy.
What's that?
They would offer surgery or
sorry, they would offer a biopsy, but they also did MRI at this just before too. But even if your MRI were negative, and as I mentioned, they really didn't do a PSA screening, they decided if you're at high risk, genetic risk, and even if your MRI are negative, we're going to offer biopsy. And there they found patients that looked completely negative on MRI. Some they did have PSA testing, so they might have been negative. A lot of more negative. They did not have elevated PSA, but yet they had clinically significant prostate cancer. So in their cohort, it was almost at the level of like 35% or more had some sort of prostate cancer. But if you're going to go by, let's say, Gleason 6 or 7 or some larger cutoff, but it was still at least, I think, 25 or 30%, right? That had. And what was interesting, though, in that study is it looked like some of our pre-screening tests missed most of the prostate cancer that was found by biopsy. So that, that's an example of taking genetic risk by itself, right, within a certain age range. I think they were like between 55 and 65, the age range, right, which isn't, it's not really old, but I mean, it's an interesting cohort because that's where you want to see if you can do a better job detecting early. And, but, it just shows how the genetic risk, as expected, right, puts you at 2 or 3-fold risk for prostate cancer, right? So if your average risk by that age is at least, 10%, and now you're at 3-fold risk, that's 30% likelihood that you are walking around totally asymptomatic without any pre-screen that's positive, right? But you have prostate cancer.
And that was April of this year, New England Journal?
Yeah, that's right. Yep.
Okay. It looks like a good one to read. I didn't see that.
So now, but in my case, it's using genetic risk first to do earlier screening, but then only doing biopsy if there's some other reason besides the genetic risk, right?
Either PSA or something.
That would be elevated PSA. Yeah.
PSA or DRE, are all those people at increased genetic risk got MRI?
Yep, they all got MRI.
Yeah,
that was part of the study, right? Because they also wanted to see could they screen or were they missing?
So that's where
they missed almost 70%. Yeah, they missed, I think, almost 70% on MRI is what they said. That's a large number. So they're going to depend on MRI to pre-screen. Then it's like, what are we doing, right? And the same thing probably with PSA, right? Because it's not really sensitive either, right? So it's just what are we really going to do going forward, right? We obviously got to try to improve some of these modalities ahead of time.
We need a better, less invasive test and biopsy. I mean, that seems like a pretty big leap, but I guess if the risk is high, it's justified.
I have a question with some of the Galateo reports that I've seen. There's 2 types of reports. There's research grade PRS and then there's clinical grade PRS. I guess this is kind of a twofold question. Can research grade PRS scores be used in the clinical setting? And is there any still, I can imagine there's skepticism if they were to use that. Right? What is the skepticism that doctors and clinicians kind of have that you guys have seen in your network to adopt PRS scores more?
So when you're talking about our company's tests, we call it the strata risk test. That's the clinical test, right, as opposed to research test, but we call it strata risk, and that covers 3 or 4 different cancers, covers, as I mentioned, 10 different cardiometabolic diseases, including coronary artery disease and LDL and hypertension and so on, right? But the research grade is more of the algorithm is exactly the same between the clinical grade and the research grade in these cases that you're talking about. But the reason we call it research grade is because we're giving the physician and the patient, ultimately, if they see the report, what exactly is their risk, right? So it's a sort of higher resolution in the sense that most of our clinical reports, we say we only report whether or not your patient is at high risk or not high risk, right? So it's that 90 percentile plus, as I mentioned, right? That puts you usually about 2 or 3-fold risk for that any of these diseases. But we don't report if your patient, and then we also give a percentile, right? So you could be at 80 percentile and not be at high risk, right? Or 70 percentile. But what we do is we have a cutoff of 60%. So if you are higher than 60%, your patient is higher than 60% at that particular disease, then we'll say precisely it's 63% or 70% or 90%, whatever. But if it's lower than 60% in the clinical grade reports, we say, we just say your patients that they're not high risk and they have a risk that's less than 60%. Because the fear has been, and the FDA has expressed this over the years as well in some interactions we had in our Decode days, even on PRSs. One of the controversies was because for BRCA1 and BRCA2, it's very easy to say you either have it or you don't. You're at very high risk, or you're just average risk, right? But with PRSs, you have a spectrum of genetic risk, as Dr. Stanford mentioned. You have a lot of patients who have protective variants, right? So if I tell you that you have a very low risk for CAD, for coronary artery disease, for a heart attack, right? Maybe you'll be tempted to continue to increase your weight or, engage in lifestyles that might put you at higher risk because you think somehow you're Superman or Superwoman, right? You are not going to have a significant disease based on this genetic profile, right? So that was the concern, that this might cause patients to, if they saw their own report, and they found they were very low risk, they may not either be screened. Let's say if you're at low risk for prostate cancer or breast cancer, maybe don't do your mammography for breast cancer because, hey, I got a 10% chance I'm going to get, or I'm in the lower 10 percentile for breast cancer, so I'm relatively protected, right? So, and that's a legitimate concern, I think, right? It all has to do with obviously the physician, the patient-physician relationship, right, and educating everything else. But just naturally, people are going to say, well, I don't need to worry about these certain diseases, right, because I'm at low risk, right? So that's why for the clinical reports, we say, okay, we're trying to go by this concept that it can be counterproductive if you tell somebody they're at very, very low risk, right? For the research reports, we've offered those same algorithm, but we actually give you the physician the direct percentile, right? Just as a way of sort of getting feedback from Dr. Stanford and other clinics, right? That can then see, is this more beneficial or more informative to give these low risk or more precise percentiles or not, right? So,
yeah, and my patients definitely understand that none of this is a substitute for the USPSTF, guideline-driven cancer screening for sure.
But I think it's
not the calorie test, not nothing. You got to start with that gold standard. What this is more information that can inform who you need to pay closer attention to for what.
Dr. Gulcher, this has been such an insightful conversation. I'm definitely going to have to listen back here because some of it was so in depth, but also like so informative. So thank you so much, Dr. Gulcher, for hopping onto the show. And if anybody wants to reach out to learn more about Galateo Bio, learn more from you, what's the best way that they can reach you?
Well, just go to our website, Galateo Bio, and you can just search for that and connect that way if you'd like.
And those will be in the show notes. So thank you so much, Dr. Gulcher. Thank you so much, Dr. Stanford.
Now, I want to thank Dr. Gulcher also for all your pioneering work. And, you really moved not just the field of genetics and polygenic risk scores, but the whole field of medicine and precision medicine forward with your groundbreaking work. Thank you so much for that.
Great. Thank you very much. Take care.
Thanks for joining us on the Precision Medicine Made Simple podcast. If this episode inspired you to learn more about your health, please leave us a review and share the podcast with a friend. Your feedback helps spread the word about the power of precision medicine. Remember, taking action today can lead to a healthier tomorrow. See you next time.